Act 1. The Opening of Arrogance, The Hook. September 15, 1943. Berlin, Germany. IG Farben Laboratories, the world's largest chemical pharmaceutical conglomerate. Dr. Heinrich Herrlein, director of pharmaceutical research, examined intelligence reports about a new drug the Allies called miraculous, penicillin. The name was familiar. Alexander Fleming, British bacteriologist, had accidentally discovered the Penicillium notatum fungus in 1928, 15 years earlier. But the substance was notoriously unstable, deteriorated rapidly, and production was microscopically small. In 1940, the world's entire production of purified penicillin barely sufficed to treat half a dozen patients. Herrlein knew the technical limitations intimately. IG Farben had dominated world pharmaceutical chemistry for decades. Germans had invented aspirin, heroin, sulfonamides. Bayer, Ernst, IG Farben were names synonymous with chemical excellence since the late 19th century. If penicillin could be mass-produced, Germans would have been first to do it. Allied reports of massive use were obviously propaganda, exaggeration designed to demoralize the Wehrmacht. Herrlein dismissed the documents and articulated what the entire German medical community believed. Penicillin is a laboratory curiosity. Fleming discovered it 15 years ago, and it still cannot be commercially produced. It requires extremely complex purification that our superior chemical industry can only do at experimental scale. Americans don't have pharmaceutical tradition comparable to ours to solve a problem of this magnitude. It's fantastical to believe experimental drug will become massively available treatment during total war. What Herrlein and the entire German medical community didn't know, what they couldn't comprehend through their confidence in European chemical supremacy, was that while they debated whether mass production was theoretically possible, Americans were already doing it, transforming British laboratory discovery into industrial pharmaceutical production on a scale that defied all German understanding of possible. Between 1941 and 1945, through unprecedented collaboration between federal government, universities, and private industry, the United States increased penicillin production from practically zero to 650 billion units per month, an increase of more than 1,600 times in just 4 years. Scientists at the Northern Regional Research Laboratory of the Department of Agriculture in Peoria, Illinois, developed deep tank fermentation that increased yield by 1,000 times. Pfizer built the first mass production plant using 7,000-gallon tanks. By June 1944, D-Day, the United States was producing 2.3 million doses monthly, enough for every wounded Allied soldier to receive penicillin. Germany desperately tried to replicate production but managed only minuscule quantities in October 1944. Too late. American penicillin saved more lives than many victorious battles. The mathematics of pharmaceutical abundance was already being written not in theoretical scientific superiority, but in production numbers so vast they transformed war medicine from improvised art into industrial system of massive life-saving. In 1942, total world penicillin production was approximately 400 million units per year, sufficient to treat only hundreds of patients with severe infections. This was all penicillin available globally after 14 years since Fleming's discovery. In 1943, American production alone reached 21 billion units, a 52-fold increase in 1 year. In 1944, production exploded to 1,600 billion units, an additional 76-fold increase. In 1945, the United States produced 650 billion units per month, more than 1,600 times 1942 world production. To contextualize magnitude, in March 1942, all available penicillin in the United States barely sufficed to treat 100 patients. 24 months later, in March 1944, American monthly production was sufficient to treat millions. The technological breakthrough came from an unlikely place, Northern Regional Research Laboratory in Peoria, Illinois, an organ of the United States Department of Agriculture, focused on agricultural fermentation, not pharmaceutical research. Scientists there developed deep tank fermentation using a strain of Penicillium chrysogenum fungus, found in moldy cantaloupe melon from a local market that produced 200 times more penicillin than Fleming's original strain. Combined with submerged cultivation techniques in 7,000 to 12,000 gallon tanks developed by Pfizer, production exploded exponentially. Each industrial tank produced in weeks what British laboratories took years to produce through surface cultivation methods in bottles. 21 American pharmaceutical companies participated in collaborative effort coordinated by the War Production Board, sharing research and patents freely, something absolutely impossible in Germany with industry fragmented by commercial competition and industrial secrets. Impact on military mortality was measurable with devastating precision. In World War I, 18% of wounded soldiers who reached field hospitals alive died of infections, gas gangrene, septicemia, bacterial pneumonia. Antibiotics didn't exist. Amputation was frequently the only option to prevent infection spread. Soldiers survived bullets and shrapnel only to die days later from bacteria. In World War II, after 1943 when penicillin became abundant, mortality from infection fell to less than 1%, a 95% reduction. Venereal diseases, gonorrhea and syphilis, that incapacitated thousands of soldiers for months and required prolonged painful treatments, were cured in days with penicillin. Conservative estimates indicate American penicillin saved 100,000 to 150,000 Allied military lives during the war, more than many celebrated victorious battles. Strategic impact transcended absolute numbers. Allied wounded soldiers had survival rates 15 to 20% higher than Axis soldiers with equivalent wounds. Each soldier saved was a soldier who returned to combat after recovery, experienced veteran kept in service. Each German soldier killed by treatable infection was permanent loss of experience and training. The accumulated difference over 4 years of war created growing numerical advantage simply through superior industrialized medicine. It wasn't just humanitarian life-saving, but maintaining effective combat force through ability to heal and return soldiers to front lines. In December 1941, weeks after Pearl Harbor, the American government classified penicillin as maximum war priority, comparable to the Manhattan Project in perceived strategic importance. The War Production Board coordinated unprecedented collaboration that violated all norms of commercial competition in peacetime. 21 pharmaceutical companies including Pfizer, Merck, Squibb, Abbott, and Lilly, universities like Oxford, Columbia, and Yale, and government laboratories like Northern Regional Research Laboratory of USDA shared research, patents, and discoveries completely. Scientists from rival companies worked side by side, exchanging information that in peacetime would be valuable trade secrets. This government-forced collaboration was impossible in Germany, where IG Farben, Bayer, and Ernst jealously guarded proprietary research even during total war. Alexander Fleming had discovered penicillin by accident in September 1928 at St. Mary's Hospital in London. Returning from vacation, he found a Staphylococcus culture plate contaminated by Penicillium notatum fungus. Around the fungus, bacteria had died, creating a clear zone. Fleming isolated the antibacterial substance, called it penicillin, and published the discovery in 1929. But he faced an insurmountable problem. Penicillin deteriorated rapidly, was difficult to purify, and yields were microscopically small. Fleming abandoned work after years trying to purify the substance for clinical use. During the following decade, penicillin remained a scientific curiosity mentioned in books, but considered clinically impractical. In 1938, Howard Florey and Ernst Boris Chain, biochemist at Oxford University, resumed Fleming's research. They developed purification methods, and in 1940 demonstrated that purified penicillin cured infections in mice. First human test came in February 1941. Albert Alexander, Oxford policeman, was dying of septicemia after scratching his face on a rose thorn. Infection spread, causing abscesses throughout his body. Florey administered all available penicillin, about 200 milligrams. Alexander improved dramatically, but penicillin ran out before complete cure. Infection returned and Alexander died. Florey had proven penicillin worked, but didn't have sufficient quantity to complete treatment. Producing enough penicillin for one patient required liters of culture grown in hundreds of bottles for weeks. Florey realized England, under German bombing with limited resources, couldn't develop mass production. In July 1941, he flew to the United States seeking help. The Department of Agriculture directed him to Northern Regional Research Laboratory in Peoria, Illinois, a research facility focused on agricultural fermentation. It seemed a strange choice for pharmaceutical production, but the laboratory had unique expertise in large-scale fungus cultivation. Robert Coghill and Andrew Moyer, Peoria scientists, attacked the problem from two angles: finding more productive fungus strain and developing large-scale cultivation methods. Mary Hunt, laboratory technician in Peoria, received the task of testing moldy fruits, vegetables, and soils from local markets and farms seeking superior Penicillium strains. For months, she tested hundreds of samples. In 1943, she brought moldy cantaloupe melon bought at local market. The fungus growing on the melon was Penicillium chrysogenum NRRL 1951, a strain that produced 200 times more penicillin than Fleming's original strain. This melon, nicknamed Mary's Golden Mold, became ancestor of all industrial production strains. Through mutation with X-rays and ultraviolet light, yield was subsequently increased to 1,000 times the original strain. Simultaneously, Andrew Moyer developed liquid culture medium using corn steep liquor, a byproduct of the corn processing industry abundant in the Midwest. This medium was cheap, abundant, and supported rapid fungus growth. Combined with deep tank fermentation with forced aeration, the method allowed cultivation in industrial tanks of thousands of gallons instead of individual bottles. Pfizer, a Brooklyn pharmaceutical company, had expertise in deep tank fermentation developed for citric acid production. They built the first penicillin pilot plant in 1942, using 7,000-gallon tanks each. The process was risky. Fermentation required absolute sterilization, precise temperature and pH, constant aeration. Contamination of one tank meant losing thousands of gallons of culture. But when it worked, each tank produced in 2 weeks more penicillin than Oxford's entire laboratory produced in a year. In March 1942, Ann Miller, 33 years old, was dying at Yale New Haven Hospital from streptococcal septicemia after spontaneous abortion. Temperature reached 41 degrees Celsius. Doctors had exhausted all options. Sulfonamides, synthetic antibiotics developed in Germany, didn't work. Dr. John Bumstead, her physician, obtained 5.5 grams of purified penicillin through contacts with Merck researchers, half of entire American production at that moment. He administered the first dose at 2:30 p.m. on March 14, 1942. Within 24 hours, Ann's temperature normalized. Within a week, she was completely recovered. First documented American life saved by penicillin. Newspapers called it miracle drug. This dramatic story transformed penicillin from scientific curiosity into national priority, proving it worked spectacularly when available. Ann Miller lived until 1999, 57 additional years thanks to 5.5 grams of penicillin. Ann's story catalyzed massive investment. Government classified penicillin as maximum military priority. The War Production Board coordinated construction of 21 production plants. Pfizer expanded from pilot tank operation to complete factory in Brooklyn. Merck built plants in New Jersey. Abbott, Squibb, Upjohn, Eli Lilly built their own facilities. Each company contributed innovations. Squibb developed filtration that increased purity. Lilly improved chemical extraction. Abbott optimized freeze-drying to create stable penicillin powder for transport. Information flowed freely between companies through government-coordinated committee. Pfizer engineers visited Merck plants. Squibb researchers shared techniques with Abbott. It was unprecedented collaboration imposed by war necessity. Production exploded exponentially. In 1942, American production was 131 billion units, sufficient to treat approximately 40,000 patients. In 1943, 21 billion units, sufficient for 200,000 patients. In 1944, 1,600 billion units, sufficient for 5 million patients. In 1945, 650 billion units per month, sufficient for 20 million patients annually. Each increase represented engineering breakthrough, new fermentation methods, better purification techniques, more plants coming online. Price fell dramatically. In 1943, penicillin dose cost approximately $20, equivalent to $300 today. In 1945, it cost less than 50 cents. Democratization through industrial abundance. Germany desperately tried to develop its own production, but fragmented and belated efforts consistently failed. IG Farben began penicillin research only in 1943, 2 years after Americans. Captured documents revealed German chemists managed to produce small experimental quantities only in October 1944, 3 years after Allied mass production began. Limited facilities at Höchst and Leverkusen were bombed before producing clinically significant quantities. Germany never achieved more than grams per month while America produced tons. Reasons for failure were multiple: industrial fragmentation with companies refusing to share research, Allied bombing destroying plants and killing scientists, resource scarcity for massive necessary investment, and fundamentally, lack of forced collaboration culture that Americans implemented through government coordination. Wehrmacht medical report from January 1945 admitted with devastating candor: captured American soldiers carry personal penicillin in individual medical kits. Every soldier has access to miracle antibiotic that cures infections in days. Our soldiers die from gangrene and septicemia that would be treatable if we had penicillin. It's not a question of German medical competence or skill of our surgeons who are excellent. It's a question of pharmaceutical industrial capacity we simply cannot match. Americans transformed British laboratory discovery into massive industrial production in 3 years. It took us 3 years just to produce experimental quantities insufficient for generalized clinical use. German physicians who treated wounded American prisoners were shocked to see each soldier carrying penicillin ampules, quantities that entire German hospitals didn't possess. Preparing for D-Day, American military accumulated 2.3 million penicillin doses, a quantity that would have been impossible to imagine 18 months earlier. Each medic carried penicillin ampules in medical kit. Field hospitals had abundant stocks. Hospital ships were equipped with refrigeration to keep penicillin stable. When 150,000 soldiers landed in Normandy on June 6, 1944, more than 10,000 seriously wounded received penicillin in first 48 hours. Estimates indicate 3,000 to 4,000 lives were saved that would have been lost to infections in World War I with identical wounds. General Eisenhower declared penicillin as important as any weapon in Allied arsenal, saving lives and returning soldiers to combat. The devastating irony was inescapable. Germans defending Normandy, wounded or killed on the same beaches, had no access to the drug discovered in Europe by Fleming, purified by Britain's Florey and Chain, but mass-produced exclusively in America. Europe invented penicillin, but couldn't produce it. America, without European pharmaceutical tradition, transformed it into abundance that saved Europe from itself. This inversion of expectations encapsulated fundamental difference between scientific capacity and industrial capacity for total war. Penicillin's impact transcended combat wound treatment. Venereal diseases were a massive military problem. In conventional armed forces, approximately 5 to 10% of soldiers contracted gonorrhea or syphilis at any time, removing them from service for weeks or months. Conventional treatments were prolonged, painful, and frequently ineffective. Penicillin cured gonorrhea in 48 hours with simple injections. Syphilis, previously requiring months of treatment with arsenic and mercury compounds, was cured in weeks with penicillin. Impact on military readiness was enormous. American Army treated more than 4 million venereal disease cases with penicillin during war, returning soldiers to service in days instead of months, equivalent to adding entire divisions of combat strength through superior medicine. April 15, 1945. Charité Military Hospital, Berlin, Germany. Dr. Ferdinand Sauerbruch, 70 years old, Germany's most famous surgeon, treated wounded soldiers in desperate defense of Berlin while the Reich completely collapsed around him. Soviet artillery constantly bombarded the city. Hospital building had been partially destroyed by bombs. Sauerbruch had supervised German military medicine during 2 world wars, witnessing evolution of surgical techniques through decades. But now he observed a pattern that transcended surgical skill and shattered any illusion of medical competence, overcoming industrial reality. German soldiers with moderate wounds that a competent surgeon could easily repair, died from infections in days or weeks after successful surgery. Gas gangrene, tetanus, septicemia killed more Germans than projectiles or shrapnel. Sauerbruch had sulfonamides, but they were frequently ineffective against established infections. He had no penicillin. No hospital in Berlin had any. Limited German production, always insufficient, had ceased completely when plants were bombed. Simultaneously, wounded American prisoners treated in the same hospital survived similar or worse wounds because each carried personal penicillin in individual medical kit. The disparity was overwhelming and philosophically devastating. Sauerbruch treated a 19-year-old German soldier with shrapnel wound to thigh. Surgery was perfect, removed fragments, repaired damaged artery, cleaned necrotic tissue. 3 days later, infection set in. Without penicillin, Sauerbruch watched the young man die from septicemia despite all efforts. Same day, he treated American prisoner with almost identical wound. He administered penicillin found in the prisoner's own kit. The American recovered completely, survived the war. 2 soldiers, 2 equivalent wounds, 2 equally competent surgeries, 2 opposite fates determined exclusively by presence or absence of antibiotic, Germany couldn't produce. Sauerbruch perceived truth that transcended individual medicine. Germany didn't lose war from lack of soldier courage, general skill, or physician competence. It lost from fundamental inability to transform science into industrial abundance. Fleming discovered penicillin in Europe. Florey and Chain, working at Oxford, purified it. But only Americans without pharmaceutical tradition comparable to Europe's transformed discovery into production that saved millions. It wasn't scientific defeat, wasn't medical defeat. It was absolute industrial defeat, revealing limitations of system that prioritized individual excellence over systemic capacity to produce abundance. Sauerbruch wrote in personal diary, words preserved in Charité archives, "I treated German and American soldiers side by side during final weeks. Identical wounds, identical surgical techniques, completely opposite fates. Germans die from infections because we have no penicillin. Americans survive because each soldier carries sufficient doses for complete treatment." Alexander Fleming discovered penicillin in London in 1928, 17 years ago. Americans, supposedly without our superior chemical tradition, transformed it into industrial production that equips every soldier with miracle drug, while we, with IG Farben and Bayer, and all our historical pharmaceutical excellence, completely failed to produce sufficient quantities. We didn't lose war just on battlefields of France, Russia, or here in Berlin. We lost in laboratories and factories that transformed discoveries into massive life-saving. Modern war isn't won by best theoretical science but by best applied industrialization of that science to equip millions. Final statistics were overwhelming in totality and unequivocal in verdict on pharmaceutical industrial capacity. In 1942, total world penicillin production was approximately 400 million units per year. In 1945, the United States alone produced 650 billion units per month, a 1,625-fold increase in just 3 years. Military mortality from infection fell from 18% in World War I to less than 1% in World War II. Conservative estimates indicate penicillin saved 100,000 to 150,000 Allied military lives during war, plus countless thousands of treated civilians after liberation. Germany managed to produce small experimental quantities only in October 1944, too late to impact outcome. After war, penicillin inaugurated antibiotic era, saving billions of civilian lives globally in subsequent decades. General George C. Marshall, Chief of Staff of the United States Army, declared in 1945, "Penicillin won the war as much as any tank, bomber, or ship. It saved tens of thousands of Allied lives and returned soldiers to combat who would have died in the previous war from now treatable infections." It was a triumph of American science applied industrially. Perfect example of how mass production capacity transforms scientific discovery into decisive military power. This assessment from supreme military leader carried weight of strategic understanding. Marshall recognized modern war wasn't won only by weapons that killed enemies, but by systems that saved allies to continue fighting. Peoria. Gerhard Domach, German chemist who won Nobel Prize in 1939 for sulfonamide discovery, wrote in 1948 in devastating testimony, "Germany discovered sulfonamides, first synthetic antibiotic, and we prided ourselves on pharmaceutical superiority. But we couldn't produce penicillin in significant quantities while Americans completely industrialized it in 3 years. They took British discovery and applied production engineering at scale that our chemical industry, supposedly the world's best, simply couldn't match. Result was, we lost more soldiers to treatable infections than Allies lost in direct combat in many theaters. This isn't scientific defeat because we knew the science. It's absolute industrial defeat, revealing limitations of system that couldn't mobilize resources to transform knowledge into life-saving abundance in time necessary to make difference. Penicillin's legacy transcended military victory to permanently transform human medicine. Fleming discovered in 1928, but discovery remained curiosity for a decade. Florey and Chain purified in 1940, but England couldn't produce sufficient quantities. Only Americans industrialized in 1942-1945, transforming impossible into abundant. The miracle wasn't scientific discovery, but production engineering that made discovery accessible to millions. After war, penicillin revolutionized civilian medicine. Pneumonia, previously often fatal, became treatable with simple antibiotic course. Tuberculosis, ancestral killer of humanity, was controlled. Surgeries became dramatically safer with prophylactic antibiotics preventing post-operative infections. Billions of lives have been saved since 1945 thanks to American industrialization of British discovery, based on Scottish scientists' observation. Historical lesson was profound and applicable beyond specific pharmaceuticals. Pure science discovers possibilities, identifies phenomena, understands mechanisms. But American industrial engineering transforms possibilities into massive reality that changes world. Germany had best chemical tradition of 19th and early 20th century. IG Farben was pharmaceutical powerhouse that invented revolutionary drugs. But Americans had something more valuable in total war: capacity to scale production from experimental zero to global abundance in time relevant to make strategic difference. This capacity applied to penicillin, Sherman tanks, B-17 bombers, Liberty ships, and countless other systems, won war through abundance that made German-specific technical superiority strategically irrelevant. Penicillin proved modern resource warfare isn't won by best science, but by best system for transforming science into products that can be distributed at continental scale. Europe invented penicillin, but America democratized it through production that equipped every soldier. This democratization of advanced technology through industrial abundance was defining characteristic of American military capacity that Germany, with all excellence in individual laboratories, never could replicate systemically. When Dr. Sauerbruch treated soldiers in Berlin in 1945, he wasn't witnessing failure of German medicine, but failure of German industrialization to apply German medicine at scale necessary to make difference in war-consuming millions.